Hello everyone, Assalamu alaikum. Welcome to my online video class. I am Dr. Marufa Akhter, Associate Professor of Biochemistry, Sakina Women's Medical College, Jashore. Today I want to have a brief outline about DNA transcription. It's one of the important topics of molecular biology and genetics. At the end of this session, we will be able to clear up about the basic chemistry of RNA, the mechanism of transcription and the processing of RNA after their synthesis, that is the post-transcriptional modifications of RNA. The genetic master plan of an organism is contained in the sequence of the deoxyribonucleotides in DNA, but RNAs are the working copies of DNA through which the master plan is expressed. And during this copying process, RNA molecules are synthesized using DNA template is known as transcription. For better understanding of the overall process of transcription, one must have a clear conception about the basic chemistry of RNA. RNA is a polyribonucleotide held together by 3' to 5' phosphodiester bases. Although RNA has certain similarities with the structure of DNA, but there are some specific differences between DNA and RNA. The pentose sugar in RNA is ribose in contrast to deoxyribose that is present in the structure of DNA. At the 2' position of ribose sugar, there is a hydroxyl group instead of hydrogen present in DNA. RNA contains the pyrimidine uracil in place of thymine in DNA. Here is the structure of uracil and this is thymine. The structural difference lies here. Thymine contains a methyl group, but the uracil does not. It is the methyl group. RNA is usually a single-stranded polynucleotide. It is a single-stranded molecule, but the DNA has double strand. DNA contains equal amount of adenine and thymine, that is base paired by 2 hydrogen bonds, and equal amount of guanine and cytosine, base paired by 3 hydrogen bonds. So, the base pairing is possible in DNA. Being a single-stranded molecule, there is no specific relation between purine and pyrimidine contents in RNA. So, it does not follow the Chargas rule, but DNA obeys it. Due to the presence of hydroxyl group at 2' position, alkali can hydrolyze RNA. DNA cannot be subjected to alkali hydrolysis due to lack of this group. Instead, DNA contains hydrogen at 2' position. But the RNA molecules are more resistant to ultraviolet rays than DNA. RNA is synthesized and processed within the nucleus, but it has to come to the cytoplasm for its work. So, its location is in the cell cytoplasm. Now, it is the types of RNA. There are 3 major types of RNA. These are messenger RNA, transfer RNA and the ribosomal RNA. It is the messenger RNA, here is the transfer RNA and this one is the ribosomal RNA. Messenger RNA comprises about 5 to 10% of the total RNA molecules in the cell, and the transfer RNA ranges from 10 to 20%. The ribosomal RNA has the higher percentage. It ranges from 50 to 80% of the RNA molecules in the cell. There are also some minor varieties of RNA. These are small nuclear, small nucleolar, small cytoplasmic, transfer messenger, and the last of all is the heterogeneous nuclear RNA. Let's have a look to the messenger RNA. It is one of the important major RNA molecule. Although it comprises only about 5%, but it carries genetic information from DNA for protein synthesis. Messenger RNA is synthesized from heterogeneous nuclear RNA using the antisense strand of DNA as template. Here is the structure of messenger RNA. It has 2 ends. This is the 5' end and this one is the 3' end. At the center, this is the coding region or transcription unit. It consists of alternative arrangement of exons and introns. Only the exons carry genetic information, so the introns must be excised. And the removal of introns and joining of exons is known as splicing. There are also some untranslated regions that do not code for protein synthesis. At the 5' end, there is untranslated leader sequence, and at the 3' end, there is untranslated trailer sequence. Moreover, at the 5' end, there is a cap. This cap is formed by attachment of 7-methylguanosine triphosphate by an unusual 5' to 5' triphosphate linkage. And at the 3' end, there is an addition of 40 to 250 adenine nucleotides known as polyadenylation. These are the adenine nucleotides that are joined at the 3' end during the process of polyadenylation. The cap at the 5' end and the poly-A tail at the 3' end, these two modifications protect the newly synthesized messenger RNA from the premature degradation by exonucleases during its journey from nucleus to the cytoplasm. Messenger RNA carries instruction from DNA for protein synthesis, so it acts as a template during the process of translation. Transfer RNAs are the smallest of the 3 major types of RNA molecules. There are at least 20 species of the transfer RNAs present in the cell that corresponds with the 20 amino acids of the protein structure. And together, transfer RNAs make up about 15% of the total RNA molecules in the cell. The structure of transfer RNA resembles that of a clover leaf. So, it has a characteristic clover leaf pattern. The transfer RNA contains of mainly 4 arms, each armed with a base paired stem. This is the acceptor arm, this is the D arm, this is the anticodon arm, and this one is the T psi C arm, and these are the loops. This is the D loop, this one is the T psi C loop, and here is the variable loop, and the last of all is the anticodon loop. Transfer RNA molecules have extensive intrachain base pairing that leads to the formation of secondary and tertiary structure of transfer RNA. It contains a high percentage of unusual bases like dihydrouracil and pseudouracil in its structure. And the presence of this unusual bases is a normal characteristic of transfer RNA. Let's have a look to the acceptor arm. This is the acceptor arm. Each transfer RNA serves as an adapter molecule that carries its specific amino acid that is covalently attached to its 3' end to the site of protein synthesis. And this arm is capped with a CCA sequence, and amino acids are covalently attached with this arm. There, it recognizes the genetic code sequence on a messenger RNA, which specifies the addition of its amino acids to the growing peptide chain. This is the anticodon arm. This arm with the 3 specific nucleotide bases is responsible for the recognition of triplet codon of messenger RNA. Here is the messenger RNA, and this is the triplet codon of messenger RNA. And the codon and anticodon are complementary to each other. This interaction is complementary to each other. This is the D arm. This D arm contains an unusual base, dihydrouridine. So, its name is D arm. This arm recognizes the enzyme amino acid transfer RNA synthetase that helps in the binding of amino acid to the acceptor arm of transfer RNA during protein synthesis. This is the T-C arm. This arm contains a sequence of thymidine, pseudouridine and cytidine. The T-C arm has an important role in protein synthesis. It recognizes the particular ribosome for the attachment of transfer RNA during translation. The transfer RNA transfers amino acid by recognizing the genetic code sequence on a messenger RNA for synthesis of protein. This video showing the genetic code sequence of messenger RNA that is recognized by the corresponding transfer RNA. Let's have a look to the ribosomal RNA or rRNA, and it is one of the three major types of RNA molecules. Ribosomal RNAs are found in association with some proteins as components of ribosome, and this complex structure serves as the sites for protein synthesis. That means they serve as a factory for protein synthesis. This is the structure of functional ribosome. It has two subunits, one is large and another one is small. The large subunit is 60th subunit and the smaller one is the 40th subunit. These are actually two major nucleoprotein complexes of eukaryotic ribosome. There are three distinct size species of ribosomal RNA. These are 23S, 16S and 5S in prokaryotic cells. In the eukaryotic cytosol, there are 4 ribosomal RNA species. These are 28S, 18S, 5.8S and 5S. The ribosomal RNAs are distributed in these subunits. The 60S subunit contains 28S, 5S and the 5.8S ribosomal RNA. And the 40S subunit, the smaller subunit contains 18S ribosomal RNA. The letter S signifies the Wedberg for sedimentation rate, and it determines the size and shape of the particles. Some ribosomal RNAs act as catalyst for protein synthesis. So the RNAs that function as catalyst known as ribozyme. For protein biosynthesis, the messenger RNA molecules must have to bind with the functional ribosome, and this binding is facilitated by the ribosomal RNA. Thereby, it helps in protein synthesis. That's all about the basic chemistry of RNA. Keep watching this video series. Assalamu alaikum. Have a nice day.